Hello, everyone. Thank you so much for joining us. I am so honored to introduce Dr. Moupali Das from Gilead Sciences. She's a vice president of clinical development, HIV prevention, and virology for pediatrics. She's a physician trained in infectious diseases and HIV with a background in academic medicine and public health. Uh, since joining Gilead, Moupali has led teams in HIV treatment and prevention, and she oversees the pediatrics team and is dedicated to ensuring efficient clinical development and global regulatory approvals to support rapid access to innovative products to help end the HIV epidemic. She is spearheading the addition of the adolescent indication to Truvada for PrEP and evaluating remdesivir compassionate use in children and pregnant women. For the past 5 years, Mopley has led the PURPOSE program, an HIV prevention clinical development, for which she has received the 2025 Manny L. Baumick Breakthrough of the Year Prize, along with her other colleagues at Gilead. And by way of Harvard College, Columbia University, New York Presbyterian Hospital, Moupali actually came to UCSF, and we're so honored that she was one of the CAPS alumna, and we're so happy to have her back here to tell us more about her research and specifically around lenacapavir. And with that, Moupali, please take it away. Thank you.
Thank you, Paria. I'm so glad to be back speaking to people at CAPS. Um, and my time at CAPS was one of my most fondly remembered times of my training at UCSF, and it's great to be here. I want this to be relatively interactive, which sometimes can be hard on Zoom, but feel free to raise your hand or put a question in the chat. I might not see it in the chat, but I would love to make sure I'm talking about things that are interesting and of relevance to you. I'm going to try to balance the background and information on lenacapavir with the initial study results. And then I'm happy to go into any of the secondary and tertiary data sets that we have presented on lenacapavir, follow-up from the studies, and additional endpoints. If that's of interest, I can also talk about um, regulatory filings, global access, and if there are any questions about career stuff, my path, other, what are opportunities for people with our respective training in biotech or pharma or in public health or other opportunities, I'm happy to cover all of that. So let me know. I would love for this to be interactive. And if you are going to ask a question, if you could turn your camera on and explain who you are as you ask the question, that would be great for me in terms of figuring out how to gear my answer. Okay, so without further ado, I will start sharing. Okay, so the talk is really on an update on lenacapavir for HIV prevention. Um, and the first thing I usually start with is to mention that this photo is a picture that I took of my mentor from college and medical school, Dr. Paul Farmer, when I was doing a rotation with him in Haiti. It's a picture of a 16-year-old boy. He doesn't really look 16. He looks younger than my 9-year-old twin boys. And he had growth delay from multi-drug resistant TB scrofula, which is when TB is in your neck. And that, led to growth delay. And then he got treated with Partners in Health and Dr. Farmer and his colleagues and grew to be almost as tall as Paul. But I just wanted to share this picture because this was a particular moment in time when I was working with Paul on a book called Infections and Inequalities. And the entire book was about how infections track along the fault lines of social inequality. And I would say in the last 25, 30 years, my addition to that is innovation without intention can unwittingly exacerbate these social inequalities and disparities. It's not a novel thought, but it's something that really guides our work. So I think everyone on the phone and Zoom would agree that there's a significant unmet need for new HIV treatment and prevention options. And what I would say is that innovation is required both in the science and in health equity to ensure progress and without exacerbating these existing social inequalities where infections thrive. So, in terms of science, in the Purpose program, it had a unique trial design, and I'm happy to talk about counterfactual trial designs with the epidemiologists and biostatisticians if they're interested at any time. I could spend hours talking about that, but that's not the focus of today. We will focus a little bit about the innovation in the product, Lenacapavir. And then I want to emphasize that none of this could have been achieved without what we call the four pillars of purpose, which are the four pillars of health equity: partnership, voice of the people, person-centric design, and a focus on including those people who are most disproportionately affected by HIV in every step of the process, from designing to conducting to supporting the trials and the dissemination of the results. So really focusing on diversity, equity, and inclusion to make that the move forward. So it's taken a long time to develop lenacapavir, and for those of you who have a background in biochemistry, um, it may be of interest to know how long it took. So first of all, what is capsid? Capsid is this rather beautiful geometric conical, it's a fullerene cone, and that has a mathematical meaning, which I can explain in a second. But it's this beautiful geometric envelope that the HIV genetic material lives in. So it's part of the virus. And each monomer that makes this capsid up is P24 antigen. So if you're doing a 4-gen antigen antibody test and you're testing for HIV, that P24 is part of capsid. And this was long thought to be an undruggable target. And what that means is it's really hard to make drugs that interfere with protein assembly rather than, attach to something and make it not work. So we started focusing on trying to interfere with capsid back in 2006. And the first generation lead didn't pan out. And the reason for that is we were focusing on stopping these monomers from assembling into the hexamers and pentamers that make the fullerene cone. We were focusing on inhibiting that. And that actually didn't work. Then some biochemist had a brilliant idea. What if we accelerate the assembly of the fullerene cone of the capsid? And that's what worked. That was our second generation lead. So we started that in 2010, and it took us until 2016 to find a clinical candidate. And if you look at the upper left-hand side, these are the least desirable molecules to most desirable, and it's really an increase in potency of the molecule. And since 2016, we've been on a fast track evaluating lenacapavir with other drugs for the treatment of the HTE, or highly treatment experienced population, who have multi-drug resistant HIV. So these are folks who had serial monotherapy or for whatever other reason have accumulated drug resistance, and lenacapavir worked very well for treating their HIV with an optimized background regimen. And so that was approved as Sunlenca in 2022. And then subsequently, actually concurrently, along with the first in human trial, we started developing lenacapavir for HIV prevention in the Purpose program. And we started recruiting participants in 2021. There was a bit of a delay because of the pandemic, and had the results last year in 2024. And since then have been working on regulatory filings and approvals. And for people in this space, I try to explain what a regulatory filing is like. And I would say it's kind of like doing, I don't know, 10 to 15 R01s for both the science, the preclinical science, the basic science, the clinical pharmacology, and the clinical data and the resistance data. It's a tremendous amount of information and really quite time consuming that requires an army of people. So the internal teams, along with our external partners who helped us conduct the studies, we have been working incredibly hard, not only during the trial, but really after the results is when the hardest work kicks in for getting these regulatory filings ongoing, because we know that it's really important to try to get lenacapavir to people who it could help most for HIV prevention. Um, okay, so this is a picture of the capsid that I was talking about. And it's a fullerene cone, so it's in the similar category of structures like Buckminster fullerene or a soccer ball. So you it always has 12 pentamers and some multiple of hexamers. And this is how capsid has multiple roles in different steps of the process for getting into the nucleus. So if you disrupt capsid and it's not working, it's hard for it to enter the nucleus. There's an impact on the nuclear porins and entry into the nucleus. And then, of course, if you can't make the proper capsid and assemble it, HIV can't make copies of itself and go on to infect other cells. So lenacapavir is very unique in that it's highly potent. It has a high stability and long half-life. And as I explained, there's a multi-step mechanism for antiretroviral efficacy. It's first in class, so there's no overlap with other existing HIV medicines. And we're studying it in treatment as part of combination therapy. We're about to launch. We've shown proof of concept that Len with two broadly neutralizing antibodies can be effective for HIV treatment with the world's first Q6-month HIV treatment regimen. In phase 2, we've demonstrated proof of concept, and now we are studying that in phase 3 trials. And we are also working on continuing to evaluate lenacapavir in different forms and formulations for twice yearly prevention, as well as once a year prevention, and looking at different ways to look at lenacapavir as a longer acting oral. So I will tell you about all of that. Okay, so we are trying to get from our current options to breakthroughs to game changers using all of the techniques that we're talking about and approaches. This is for our entire virology portfolio. So trying to get in the PrEP space from twice yearly to once yearly, and from daily to monthly or weekly. So some of you may have seen this tree slide, as we affectionately call it. It's an acacia tree, and what I tried to do was represent that the purpose trials really grew from the roots and the foundation of the 4 pillars of purpose, those principles of health equity that I refer to, as well as the robust work in treatment that was done by my colleagues prior to us initiating the purpose program. So I'll tell you about the results from purpose 1 and purpose 2. Purpose 3, 4, and 5 are ongoing, and we expect results next year, and I'll talk a little bit about those if we have time. So nothing about us without us. We started talking about the purpose trials in December of 2019. We had 2 community engagement meetings. One was in New York City, and this one was in Kigali, Rwanda. And at this meeting, we brought together civil society, advocates, people living with HIV, people using PrEP, ethics people, investigators, site staff, government officials, all to think about and global HIV activists from all over Africa and also in the United States, to think about the best way to design these trials. Sorry, I'm losing my voice. And one of the things that I wanted to know is how people felt about including adolescents, so 16 and 17-year-olds, in the adult trials, which had never been done for pivotal trials before. I also wanted to know how people felt about our proposed pregnancy policy. So, because I wanted to know if people thought that it would be safe and appropriate to include pregnant women and lactating women in our clinical trials. And there was a lot of discussion. And there were some people who felt incredibly uncomfortable with including pregnant people in the trials. Very, very uncomfortable, despite all of the evidence that we had from the preclinical space, as well as, years of experience with Descovy and Truvada and HIV treatment, hepatitis treatment, et cetera. And so there was a big back and forth, as community meetings can sometimes, or stakeholder meetings can sometimes be. And a young woman stood up, and she was actually 17 years old, and she was quite pregnant, and we hadn't noticed that. I had not noticed that because we had been sitting around a conference room table. And she said, I want to be able to be part of this trial, and I want you to include people like me and my friends, and I don't want to get HIV myself. I don't want my baby to have HIV, and we should have an ability to be in these clinical trials. So her story, honestly, was more helpful to me in changing hearts and minds internally and externally, in allowing us to have our incredibly rigorous and safe process for including pregnant people and lactating people in our clinical trial. It also allowed us to get data on pregnancy, lactation, lenacapavir levels in breast milk and in infants in people who reconsented for that process, approximately 8 to 10 years earlier than we normally have that data in antiretrovirals. So I often get asked the question, like, how do we pull this off, from people who have done a lot of interventional drug trials and know how challenging it is to include adolescents in adult trials and include pregnant people, and not just kick people off study drug and out of the study, which has happened, for very good reasons, because people are worried about impact on fetuses from experimental and investigational drug products. But there is a way to do it safely, and we followed it. And there have been numerous articles written about what you need to do to follow this approach in clinical trials of new drug products. And I'm happy to talk about that in more detail if people are interested. Sorry, now we went backwards. Okay. So that meeting kicked off the community engagement with our stakeholders, with government officials, with civil society. And the meetings across the top are the meetings that Gilead sponsored. And we established global community advisory groups for each study in Purpose 1 and Purpose 2 that were representative of the populations that we wanted to meet. And once we were out of pandemic flight restrictions, we started going and meeting with folks in person, and also bringing people together in California to meet once or twice a year. And then we met with the different community advisory groups every other month. We also continued to have robust collaborations and attended meetings sponsored by others, including AVAC and other stakeholders, and continued to meet with folks who weren't on our community advisory group to get advice about how to conduct the trials, how to do the trials, how to set up plans for access. These are some of the things, some of the pieces of feedback we received, and we took all of these to heart and have actually done all of these things. And there's abstracts on each one of these little thought bubbles that have been presented or described in abstracts or publications. So let's get a little bit into the data. And I don't see any questions for anybody, but if people are totally familiar with the data, I don't have to go through the initial results in that great detail. What I will point out is that lenacapavir and FTAF were both evaluated in Purpose 1, which took place in cisgender adolescent girls and young women aged 16 to 25 in South Africa and Uganda. The trial was stopped early for efficacy by the Independent Data Monitoring Committee, and that occurred when 50% of the participants were through week 52, the primary endpoint. So even with half the people through the primary endpoint, we were able to see tremendous efficacy with lenacapavir. And the other thing I just want to point out to people is lenacapavir's dose is 2 injections subcutaneously, along with 2 pills of lenacapavir on day 1 and day 2. Those pills are critically important because lenacapavir is a long-acting product, and it takes a while to get up to the high levels of concentrations that are required for antiretroviral efficacy, antiretroviral efficacy, and that's probably between 21 and 28 days. But if you take the oral pills day 1 and day 2, you are protected 2 hours after the day 2 oral dose. So the average person is protected 2 hours after the day 2 oral dose. And I think that's really important for people to understand the importance because people are often going home with their day 2 pills. So for the clinicians and providers in the room, that's one way to emphasize the importance of taking those oral pills at home. So, and then Purpose 2 had the same trial design, except for it did not include a Descovy or FTC TAF arm because that drug has already been demonstrated to be efficacious in cisgender men and gender diverse people in the Discover study. This program was one of the most gender, racial, age diverse, ethnically diverse, globally diverse PrEP trials conducted. You can see in Purpose 1 where most of the participants were from, and also in Purpose 2, we had folks in the United States, Mexico, Brazil, Peru, Argentina, South Africa, and Thailand. And I do think it's important to point out that Purpose 2 also had participants in South Africa. Okay, so here are the baseline characteristics. I know that these two data tables side by side are quite dense on this slide, so I just wanted to point out a few things. So Purpose 1, which was limited to 16 to 25-year-olds, had about 100 plus people who were 16 to 18 years old. In Purpose 2, the range was 17 to 74, which I think is quite interesting with the number of people in the older above 50 group being relatively significant, especially in the United States. And then we had about a third of people in the young age group, 16 to 25. There were high rates of sexually transmitted infections in the groups randomized in the clinical trials. So these were people who were screened at baseline and obviously treated through the trial. There's a high rate of chemsex in Purpose 2 and an interesting, you know, in the 16 to 25-year-olds, not a high rate of prior use of PrEP. And, only about a fifth to a quarter of people had prior use of PrEP in Purpose 2. So I think that's important to emphasize because we really brought in people who weren't currently on or had used daily oral PrEP in Purpose 2. So here are the results. There were 0 infections in Lenacapavir in Purpose 1. The amount of infections in FTEF and Truvada, so Descovy and Truvada, were similar to the background HIV incidence, which was estimated using recency assays and the recent infection testing algorithm. Happy to talk about that with anybody who wants to later. But the number of infections were really similar. I didn't mention this, but the trial was randomized 2 to 2 to 1, Len to FTEF2 and FTDF1. So approximately similar infections in FTEF and Truvada. No statistically significant differences across these three, but highly statistically significant for Len, obviously. And then in Purpose 2, there were 2 infections, over 1,900 person years, and fewer infections in the Truvada arm, relatively in comparison with an incidence of 1.7 versus 0.9. But those are the primary efficacy results. Here they are represented in terms of the rate ratios. So there was 100% efficacy for lenacapavir in Purpose 1 and 96% in Purpose 2. And the comparisons to Truvada for oral PrEP, which was the secondary endpoint, was 100% in Purpose 1 and a reduction of 89% in Purpose 2. So what happened with oral PrEP? Do the Purpose 1 results mean that oral PrEP does not work in women? I don't think that's true. I think the answer really lies in adherence. So in Purpose 1, this is dry blood spot data. So in a 10% representative randomly selected sample, we looked at adherence using dried blood spots and tenofovir diphosphate levels. People who are familiar with P. Anderson and other folks work with this will know that high is 4 doses or more per week, yellow is medium, 2 to 3 doses, and low is less than 2 doses per week. So you can see by the end of the study, 93% of people were having less than 2 doses per week. Whereas in contrast, the rate of on-time injections was 91% and 94%. And that included both, because these were blinded studies, Lenacapavir as well as placebo injections. So really high adherence to getting the injections and coming back for those on time, much lower adherence to the pills in Purpose 1. And here is the results for Purpose 2. So in Purpose 2, people did start off with more adherence to oral PrEP, and this may be due to the differences in the population, but adherence did decline over time, but still much higher than in Purpose 1 with Purpose 2. And sorry, the rates of adherence to the injections was above 90%. So the injection adherence was very similar in both studies. So in contrast, with FTAF and FTDF or Descovy and Truvada, there's really high adherence. If there's really high adherence, we have high efficacy, and this was seen in the Discover trial. So here on the same numbers, the same legend for number of tablets per week, you can see that there was much higher efficacy with FTAF and Truvada in Discover with fewer infections because of the higher oral adherence. So we did a case control study to look at Descovy, comparing Descovy or FTAF to Truvada, and we looked at the people who got infected versus the people who acquired HIV versus the people who remain negative. And you can see among the people who acquired HIV, there were many more people who are taking lower amounts of oral pills of Descovy. And then on the right-hand side, I have the comparison to Discover, which was our trial where we evaluated Descovy and Truvada for HIV prevention in cisgender men and gender diverse individuals in mostly gay men and a very few small number of trans women in that study. So the bottom line is we understand with both Truvada and Descovy, there's an adherence efficacy relationship, and the odds of HIV acquisition were approximately 90% lower in cis women who took Descovy more than 2 pills per week. And that's a very strong statistical relationship, and I think provides support for the recent IAS USA guidelines that did just recommend Descovy for cisgender women who aren't able to take Truvada. And that is not in the Descovy label, just to be very, very clear, but it is a guideline recommendation. Okay, so we included pregnant people in the studies. We had many pregnancies. Some people got pregnant more than once, which is why the number of people with pregnancies is lower than the confirmed pregnancies. And the expected rate of pregnancy outcomes was very similar. The rates across the arms in the study and compared to the background expected rates were very similar. So we didn't see higher rates of adverse pregnancy outcomes than expected for populations of young women in these geographies. Just going to pause a bit here. And for people who conduct clinical trials, especially globally, you may realize that we're under a tremendous amount of pressure to conduct these trials in a ethical manner, and that's very critically important to us. On the other hand, sometimes the ethics committees and IRBs or internal regulations at sponsors can limit some of the resources that we're able to provide in the trial because folks are very concerned about undue enticement. So this slide is a set of pictures of where the women are coming across Lake Victoria in these wooden canoe boats with speedboat motors to deliver at this labor ward and this maternity wing of the labor ward, just to show you where we're working with. And when women were coming over, Um, One of the challenges in some of the locations in Uganda is that people are required to provide their own gloves and delivery supplies because the labor wards are not well resourced. So the onus is on the pregnant person and their family to provide these resources. And if they don't have the resources, they cannot be attended to because the healthcare personnel do not have gloves or other supplies to take care of them in a safe fashion. So our Ugandan investigators and site staff had asked us to supply these resources for the people who were pregnant in the clinical trials. And it took a lot of discussions internally and externally to get this approved because there was fear that we were providing undue enticement to the pregnant persons and giving them extra resources that we weren't giving to other people. And there were all sorts of concerns about this, but we were able to help explain the situation and explain why these resources were required for people to be safely included in our clinical trials. And eventually, after lots of discussion, we're able to provide this mama kit, which was also pioneered by WHO in regular implementation, but we were able to use the mama kit in the clinical trials to provide support in Uganda. And on the right-hand side, with their permission to their investigator are some of the purpose moms and babies in this Ugandan site. Okay, there's a lot of firsts in the purpose program, and I've alluded to a few of them, really with this theme of focusing on science and health equity. And here we have some of the innovations in science and what we did there, and then innovations and how we did health equity. And we feel that both of these pillars were critically important for achieving the unprecedented efficacy and inclusion in the clinical trials. Len and the two oral tenofovir PrEP prodrugs were safe and well tolerated in both of the studies. These are the adverse events excluding injection site reactions, which I will talk about at a later slide. Um, Thank you, Christina, for your feedback. I'm sorry, I haven't been checking these questions. Please feel free to put them in the chat and I'll move this over so I can see it, or raise your hand. Um, I think the only difference by arms was really in creatinine clearance and renal abnormalities in Purpose 2. But that is a well-known impact of tenofovir, and so a really well-tolerated drug. I'm going to talk a little bit about how Len works. So lenacapavir as a subcutaneous injection forms a drug depot. It needs to be injected in the fat layer or the subcutaneous layer that's underneath the skin and above the muscle. So this is the muscle, this is the skin, and these little red ovals are meant to indicate how tightly dense the skin layer is. So you need to be in the fat layer, and the ideal place to inject lenacapavir is where people have a little bit of subcutaneous cushion, if you will. And the injection technique is really critical for injection success. So pre-injection, we found ice to be incredibly helpful for numbing the layer of skin and reducing the initial injection site pain. Um, it's really critical to try to inject at a 45 to 90-degree angle, and you can do a gentle pinch to help the injection go into the subcu space. Now, if somebody has totally flat belly, the abdomen region also extends into the intraaxillary line, and like some people may have love handles even if they have a really flat belly, so look for the place with a little bit more cushion to give the injection. And for those people, you may need to be to the closer to a certain angle of injection to make sure that you're not hitting muscles, so you may not be able to go full 90 in some people. And then post injection, you want to make sure you're giving ice too, as well. So in the injections that I've seen administered in the clinical space, post-approval, people just slap on the ice packs while they're talking to the patient, doing the informed consent, pulling up the injections and preparing themselves. And then by that time, the ice has been on the person's skin for approximately 5 to 10 minutes or longer, and that helps with the initial injection site reactions. So the next slide is purpose one and purpose two injection site reactions. And let me explain what this graph is. So green means no injection site reactions at all. Yellow is a grade 1 injection site reaction, and grade 2 is red. The rates of grade 3 were so small that we couldn't graph them, but they're very small numbers of grade 3 injection site reactions. All the data is in the Yes to Go label, which I'm happy to provide a link to if you want more numbers. I think it's important to note that the most common injection site reactions in purpose 1 were nodule pain and swelling, and in purpose 2, nodule pain and erythema. 99.9% of people in purpose 1 were of Black race, and therefore it would have been a little bit harder to see erythema, but there were importantly no keloids and no other challenges in terms of injection site reactions. Nodule is most common because lenacapavir is a drug depot medication, so it forms a nodule, and depending on people's body habitus and the amount of sub-Q fat they have, you can feel that nodule. Usually can't see it, but you can feel it if you gently palpate over the skin where you know where you gave the injection. Okay. All right, so I'm going to pause now and take a vote on what people want to hear about next. So I can talk about more clinical data. I can talk about our access plans. I can answer questions. I have a lot more information with more data. So if people have questions, I can go through that. So I really would love for this to be more interactive. So let me know. Christina.
Hi, Moupali. Do you have any info on management of leakage? Because both clinically in our own practice, I mean, when it's in Sun Lanka, we feel like we don't have to worry so much because there's a background regimen, but obviously for Yes2Go, we're going to be super concerned if we're going to lose drug that we're going to put the patient at risk for acquisition of HIV for lost medication. And also in the PrEP line, we've gotten a couple of cases that is asking the very same question. And I know that, Gilead has been very accessible to folks with those questions. And so far they're like, you have to make a clinical judgment. Like, how do you, it's sort of, I think what's like just not very satisfying is sort of like, how do you know how much leakage is enough leakage that it warrants like redosing? But
Yeah,
we may not even have that information, but I just wonder if
right.
you had any other like comment on that.
So my first comment is line is yellow. And it's really obvious when it leaks because it will stain clothes, it will stain, 4x4s or 2x4. It will stain because it's yellow in color naturally. So sometimes the amount of concern about the leakage exceeds the quantity of leakage, if you know what I'm saying, because it looks a lot more worrisome than if it was a clear solution and you didn't really notice it, especially if it turns somebody's clothes or the dressing or whatever, the Band-Aid yellow. So that's my first piece. My second piece is how much leakage is enough to worry about is kind of unknowable because we don't have enough infections to know about failures, right? There's only been a total of 4 in the entire program over the last 5 years. So it's really hard to know like at what like the only thing that I think we could do to really understand it is if there's leakage, send a Len level and figure out what that Len level is, but you would have to check it immediately and then at 21, 28 days and periodically onwards. Like we'd have to do like a clinical study of like a cohort study of enrolling people with leakage. But given the fact that there have been instances of leakage and they were not associated with the infections that occurred, so a lot more people had visible yellow drainage or a combination of Len with, maybe some serosanguinous fluid, which could also look yellowish or like slightly reddish. and none of those people got HIV, I think is very reassuring. I would step back and make sure that there's proper technique. So especially for hotline or warm line calls, and with people who are inexperienced in delivering the injection, make sure that people are availing themselves of our nurse educator resources, where the nurse educators we have an army of nurse educators who can go in and help people um, learn how to do the injections properly and get a lot of like practical experience prior to implementing. And sometimes a refresher may be helpful as well, because the more time we've been using lenacapavir, we've learned more about it. So there's a video online, there's diagram pictures, but then there's hands-on teaching available, obviously for free from us, on how to do the injections well. I don't really know how to answer like when we have to be concerned or not. I would just encourage people to document how much, like what amount of fluid was lost, and counsel them that it might look worse than it is because of the color of lenacapavir being yellow. So it's just more obvious than if you have leakage from insulin or Ozempic or other subcutaneous injectables. I don't know if that's helpful. There is no official kind of answer other than to like monitor people very closely. And then, we're not currently doing any uh, post-marketing surveillance or cohort study on this particular topic that's above and beyond our regular post-marketing surveillance. So if there is significant leakage, they should also call and report it. So Gilead is aware of how many cases are happening in the post-marketing setting. Okay.
Thanks,
Great.
Moupali.
Thank you so much.
Jonathan, Orlando, and I actually did the nurse training, so it was actually really helpful. There's several questions in the chat, starting
Okay.
with access plan from Carlos, and a bunch of people plussed it, and then questions on implementation gaps
Yeah.
due to
Okay. So why
HIV
don't I go
prevention.
through access first, Parya, and then I'm going to do a quick round of clinical updates over the last several conferences since we did the primary results. How about
And I
that?
think you need to come back because we have a lot of questions for you.
I'd be delighted. You guys are my favorite people, my people. Okay, let's go back. All right. So actually, I'm going to skip to access and then come back to the clinical data. Okay. So let's start on this slide. Okay, so our first approval was 6 months after we filed. We got breakthrough designation and priority review. I just like to highlight that usually it takes 8 to 10 years to get data in pregnancy, lactation, and infant or other pediatric PK and ART labels, and we were able to do it with the first approval. This is something that required a lot of banging my head against a wall and changing hearts and minds, so we're incredibly proud of this. Here's our global filing strategy. So the first step towards access is making sure you have regulatory approval. So we filed in the United States and were approved in June of 2025. Then we filed in Europe. And then one thing that's important about the European filing is not only did we do European Medicines Agency or EMA, we also did a process called EU Med for All. And EU Med for All is a process by which low and low middle income countries who have less resources, period, including in their regulatory agencies, are able to use the European filing and the WHO collaborative registration process to quickly approve the drug in their countries because they're relying on the stringent regulatory authority approval. So once we got the CHMP positive opinion in July, that kicked off the WHO pre-qualification process, and our WHO CRP countries are listed here. This list of countries was our first chunk of countries where we were filing with the WHO CRP process. And this list was curated with a tremendous amount, like 3-4 years of engagement with experts in the field to figure out which countries should be in this priority list, with lots and lots of advice from advocates and important stakeholders globally. Okay, so then next, We filed in some of the other regions that are critically important. And then we have our approvals. So blue, South Africa, Zambia, Zimbabwe are the places that we have, and Malawi over here are the places we have approval. And then dark gray is where we have filed in the WHO CRP countries. Our goal is to file in Argentina, Israel, Mexico, Peru, Saudi Arabia, and the remaining countries on this list by the end of the year and or the beginning of next year. Our team has been incredibly busy. Each one of these filings, I really think, are equivalent to about 10 to 15 R01s. It's a tremendous amount of work, and each country has their own nuances and quirks, so everything has to be tailored and customized for the particular place that we're filing. But this WHO CRP process was tremendously helpful. Zambia was approved in 19 days. 19 days. That is unheard of. And it's quite unheard of to have approvals in sub-Saharan Africa the same year that we have approvals in the United States and Europe. So we're incredibly grateful to all these people internally who've been working on this, along with all of our external partners. But we know that innovation in science and health equity, all of that is not enough to get to access. So not only do we need the simultaneous filings, but we need other ways of doing things. So the first thing I want to highlight is that we did voluntary licensing to generic companies, and we shared our intellectual property even before we filed in the United States. We transferred that technology and we picked 7 generics, but we know it takes about 2 to 3 years to get the generics up and running, so they're bringing down price. So for that reason, we also made a ton of supply to give 2 million people enough lenacapavir for 3 years. And we have formed a ton of agreements that I'll show you, multilateral agreements to get that supply to the people who need it the most. And the other thing I'll highlight is there's a QR code here and this link of our access page on our website. And I would recommend going to it because these slides are out of date like every time I present. I've been presenting almost weekly and I have to update my slides for the updated approvals, the updated filings, and the updated agreements. So you can see here, this is when we had our efficacy data, and this is when we signed the voluntary licensing agreements, right? Like 2 weeks after we saw the Purpose 2 results. We were waiting for those because we didn't know if it would work in all populations. Then we transferred the IP and technology and months before the approval in the United States or Europe. And we got Len to Eswatini, which Eswatini and Lesotho come with the South Africa approval. So we got Len to Eswatini just 5 months after US launch. And this is our manufacturing folks based in California, and some pictures of getting the drug to where it needed to go. And this is health ministers and folks from Global Fund and our team bringing lenacapavir to Lesotho, which happened last month, and told you that the data for 3, 4, and 5 would be available in mid-2026. I see a bunch of questions, so I'm going to stop sharing and answer those questions. Okay, where are my...
Well,
Okay.
just in line with the access plan, is there any updates for the Caribbean and Mexico?
Yeah, so Mexico is on the list. It's going to be happening soon. We are global teams are actively going to be filing. I don't know specific Caribbean countries and when they're on the list, but this is wave 1 of the high priority countries as determined by donors, funders, community advocates, civil society, and governments that we engage with over the last 3 to 4 years to come up with this wave of plans. But we intend to file in as many places that Gilead can file directly because that happens a lot faster than generics filing. So our goal is to file in all the places where the burden of disease is the highest and people need lenacapavir the most. Yes, there's also a new RFP that was released on World AIDS Day. So for people interested in implementation science in the countries where Len is being supplied, important implementation science questions, you can check out the RFP and apply to us. Um, there was so at Gilead, we are using this pregnancy policy in many of our clinical trials across virology, including in HIV treatment, as well as prevention as appropriate per the product and the degree of information know with the product. So we are very excited to be helping our colleagues use those do those approaches to include pregnant and lactating women. Um,
Hopefully, there's a question from Kate Haas about what you think about the key implementation gaps for Len, given that so much has been learned from Purpose. And then a
yeah,
question from
so
Judy
right
Arba.
now let me show you our forest slide. Sorry. Let's so this is where I'm gonna just keep it not in presenter mode and try to make it as big as possible. But right here, these are where we're doing our implementation science studies. Kate, there's a lot of critically important questions about access and uptake. What are the barriers and facilitators? How can we share lessons learned from early adopters to later adopters? So we've worked in the United States, we're working with Adolescent Trials Network, and we have our own inclusion portfolio as well as our own phase 4 study of Propel. And then for 3B studies, we're working with Unitaid, with ImPrEP Brazil and Virology, and then Gates with Align with Linda Gale. And we are also have released this global inclusion RFP, which is active now. We do have a once yearly formulation that is in the field. Here's the study design. So we have our purpose tree, our implementation science tree, and then Purpose 365, which is a smaller study of the 12-month free acid formulation, which is a once in a year intramuscular injection. So I will take this down. Okay. What would I have done differently if I had NIH or other public funding? Judy, I think the issue with the mommy pack and inclusion of pregnant people would still be an issue with public funding, to be honest, because the hardest people to convince are ethicists and IRBs, and that's present regardless of the funding source. And there is more of an onus if you're doing an interventional trial because of concerns about that. But the rules around undue enticement and how much reimbursement people can get is set by local IRBs in most of these countries, as you fully are familiar. So a lot of it was negotiating with them that we weren't treating the pregnant people separately, but we were giving them resources because it was required for their safe pregnancy and delivery, if that's helpful.
Yeah, I was mostly thinking, were you able to do more than you thought you could have done if you were federally funded, being funded by a private entity instead?
Honestly, having worked in both situations, I think there's more freedom in doing what we want in private situation in biotech and pharma. If we're talking clinical trials, interventional clinical drug trials.
Thanks.
Okay.
And Moupali, I'm sorry, Kate Koss also had a second part to her question. She was interested in the future of HIV prevention trial designs and what you learned from
Yes.
including background incidents.
So, Kate, Lillian Brown actually gave another CAPS alumna and UCSF ID person gave a great talk in Rwanda about the different counterfactual estimation techniques, and I'm happy to share that. It's actually all of our presentations are at Gilead Sciences Ask Medical conferences. You can find every presentation we've ever given, but I can share it with you as well. I think that there is a tremendous future for using counterfactual and background HIV incidence designs for future trials in prevention, in helping reduce the trial sizes required, but it has to be done very judiciously and carefully. And there's a reason why the other sponsor chose not to do it for their trial, because their initial version of how they implemented the trial, their background incidence was lower than the incidence on Truvada in many trials. So there was their implementation of the recency assay and their process of doing it was very different than ours. And I think ours yielded a higher background incidence estimate. It is conservative because there is some amount of selection and channeling bias of who, like, you would not send a positive person into a clinical trial for HIV prevention. So there is some impact of that. So I think that that's a particular challenge. But I think it should be used for new vaccine trials. Okay, starter kit. Yes, Christina, it's very hard, and it's different in every insurance company, every state, every local jurisdiction. So we're trying to make it that people get the injections and the oral pills together, but it depends on each person and their insurance situation. So that is that. Uh,
Actually, I just wanted to also say for any all the early career and later career folks who are interested in hearing more about a career in biotech and pharma, Moupali has kindly agreed to be able to speak to you individually and separately. So please feel free to reach out to her. And definitely, Moupali, we would love for you to come back, please. This is
Sure.
so
And if
enlightening.
people have a few minutes, I will just give you some tips, like high level tips. And this is early and mid career. For people who are interested in pivoting or exploring other options, the number one recommendations are make sure your LinkedIn is up to date and you have a accurate picture. And if you're interested in a particular job, think about it as writing your LinkedIn profile like you're writing a grant application. Look at what the employer is interested in and make sure you use their keywords, because everyone is using AI to screen. So you need to explain your experience using the keywords that the employers are looking for. And you can have different profiles or different short form CVs for different jobs for that reason. Do not send your 25 plus page UCSF CV. Make sure you have a short form CV, especially, and that it describes your work in a way that is easily translatable to what the required and preferred qualifications are in the job description. So those are my two tips that seem kind of obvious, and if they are, sorry. But those are other things, but I'm happy to talk about career opportunities in public health, having worked at New York City and San Francisco, as well as potential opportunities in academia, in non-academic positions. So good luck to everybody, and thank you so much. It was so fun to speak to all of you, and maybe next time I'll come in person because it would be great to see people live.
Yay, thank
Take
you
care.
so
So much.
much.
That's amazing. Thank you.
All
Thanks
right.
everyone who
Thank you.
joined.
Thank